All right, so how to mimic nature. That was the topic of the discussion. And I think that from my point of view, I am really interested in self-assembly processes and molecular design, structure-function relationships. And I think that was really elegantly discussed in Patrick's presentation on the intimate role that structure has with function, especially in energy harvesting. My area of interest primarily is in the realm of health sciences, and we'll talk about some of the big questions associated with that, hopefully in a minute. In a former life, though, and I did this on purpose, hopefully, because I had a feeling your talk would talk about some elements of structure and function of light-absorbing units. While I was at MIT doing my postdoc, I started to get interested in phycobalazomes and photosynthesis as well. What we see here is, I'm hoping will sort of blow your mind, all right, with respect to the intricate nature of self-assembly in biology, all right. So what we have here is we basically have a self-assembled system composed of different donut-shaped molecules indicated here by these vertical lines that stack sort of as tubes that align preferentially so they absorb light and transmit light across the spectrum from here into the core. These cores are actually tubes as well that project out of the TV plane here. They radially orient, okay? Embedded in each one of these phycobalazomes, we can see here the actual structure itself, right? There's actually two parts. We'll see that in a second. But each of these is actually a protein. So this is one protein, and this is one protein, and this is one protein that self-assembles, all right? Embedded in this matrix, as Patrick sort of alluded to or discussed before, is the actual light-absorbing units, okay? And we see here that each of these donuts that stack on top of each other, which would be a dream for Tim Hortons fans, each of these donuts is actually composed of two maybe not identical, but two different rings that actually sit on top of each other. So and I mentioned already that each of these rings is composed of three different proteins. And so there's 6 proteins in total that have to arrange and then dimerize. And then these donut-shaped entities actually link through this hole by specialized proteins, as illustrated here sort of really poorly, with the donut below them, okay? And all of this is so that the spectrum of wavelengths that hit the antenna are able to be harvested and transmitted to the core, which then allows for harvesting and transmission into the membrane, where photosystems sit and convert all the energy into chemical energy, as Patrick already really well described. And all of that pretty much exists for the sole purpose of maintaining orientation and of these chromophore units, as illustrated here. This is all the proteins have been stripped away. And basically exists as a matrix to hold these molecules in certain orientations so that they can link with molecules or chromophores below them so the energy can be transferred down that chain, which is a fascinating illustration of self-assembly. And you might realize or come to the conclusion then that self-assembly is not just the same as flocculation or aggregation or something like that. Self-assembly has this innate attribute of structure, all right? Like some sort of structure is formed due to self-assembly. So that was my energy part, and I'm going to move into health sciences a bit more now. So the question that we've been looking at for a while is understanding short peptide self-assembly. And the reason for that is, there's multiple reasons for that, but the primary reason that we're interested in is peptide self-assembly allows us the ability to modulate the peptide chemistry and structure, and then see how that reflects itself in the macro scale, perhaps, or at least in the nanoscale architecture. So what we have here is sort of an illustration of sort of a well-known self-assembly protein, RADA, the dimensions of which are provided here. They self-assemble spontaneously into nanofibers. These nanofibers, when in high enough density, form physical cross-linking that can support its own weight to something around, to make hydrogels of around 99.5% water. So if you just think about that for a second, this droplet here is almost 100% water, but it has enough ability to maintain its shape due to the 0.5% of the protein that's within it. And that's primarily due to the fact of the structure that it forms at the nanoscale, all right? So these peptides have been studied for a while in some ways. They've been shown to be relatively biocompatible. They have a flexible molecular design. We have a PNAS paper where we looked at large and small protein release through these matrices and trying to understand the effects of the peptide chemistry on the diffusion through these substrates. But what got me interested was this water capability, that these peptides were able to actually carry that much water and associate that strongly with that water. And so we started to look at just small tweaks to the peptide chemistry backbones, where we put lysines and serines on the end. And lysines with a positive charge, serines have an OH, so for hydrogen bonding. And we started to investigate freezing water versus non-freezing water. So non-freezing water, for example, is water that does not go through a freeze-thaw phase, right? So it's tightly bound to the peptide nanostructure or the peptides itself. And so when we started to do this, we noticed that just by adding these small peptide end groups, that we could change this non-freezing water content. And when it came to K5, it was relatively, on average, increases as a function of how much K5 was introduced to the system. With respects to the S, or the serines, we didn't see that. It's basically stayed similar. And so we started to think about, well, how does this affect peptide self-assembly? And so we got interested in thinking about water structuring around these nanofibers with these end groups. And we were actually able to tweak out experimentally that RADA, the base self-assembling molecule, actually dispersed or displaced about 25 molecules of water per peptide as it self-assembled, whereas the serine actually incorporated 29 molecules of water per molecule. And so we were able to show that for some of these peptides, hydrogen bonding had a role. We were able to show that commonly held beliefs that electrostatic interactions dominated the self-assembly of these molecules, that in fact they were not as important as, say, hydrophobic and hydrogen bonding attributes. And so that's just sort of, the scope of this work really was to give you, 3 or 4 case studies of how we're trying to mimic nature. And so the first study we looked at just now was primarily revolved around how does peptide chemistry at the molecular level affect nanoscale architectures and maybe even internal morphologies of hydrogels and et cetera. And now we'd like to take that one step further, because we're really interested in taking these and using these motif changes to study how they might affect not just self-assembly, but all the way up to cellular effects. So how can we tell a cell within the matrix to do something? How can we give it peptide mimics of maybe proteins or things determined from phage display assays, et cetera, to get cells to activate or to heal or to wound heal or to whatever the case might be as we want. And as an engineer, that's sort of what gets me up in the morning. And so mast cells is a really cool application of this, because at the inflammatory response for humans and other animals, obviously, mast cells play a critical role as an effector cell. And as you can see from this illustration, they play a role in everything from neurotransmission to angiogenesis, to wound healing, to matrix degradation, matrix rebuilding, and they initiate all of these processes. So they have a multifaceted they're a multifaceted chemical reactor that if you give them the right chemical knowledge or signals, they can respond in a way that you want them to. And so the really interesting thing about these is that people have problems studying them in mature states. Moreover, because of the complex nature of biology, the matrix that they reside in is not really well understood. And so with the building of an artificial matrix through self-assembly of peptides, we can really start to systematically vary chemical signals for the cells to respond to. And we can start to understand about synergies of different molecules. We can start to understand how maybe these synergies start to affect cell signaling pathways. And then we can start to think about how adding other cells to the matrix may affect everything. So the ultimate goal here really is artificial matrix development that we can inject into wound sites to direct endogenous cells to do what we want them to do. So it turns out that this RADA sequence, this RADA, is a mimic of RGD, which is a relatively well-known sequence for integrin binding. And so we tried right away just to see if we could get mast cells to culture on the matrix directly. And it was found that in fact we could. Not only did they adhere to it without stimulation, we saw that they started to migrate into the matrix. And all the while, it didn't actually induce any activation of the cells themselves. Moreover, we started to see that these matrices, as the mast cells were put into the matrix, and then we started to actually induce some sort of response, whether we did it through an IgE modified or an IgG-induced stimulation or some chemical stimulation. We saw that the matrix actually absorbed proteins that the cells were starting to produce themselves. So it's really acting like a matrix, right? It's interacting intimately with the cell, and they're starting to talk to each other. The cell is starting to build its own, ECM analogs, extracellular matrix analogs, and they're starting to build a communication network chemically, basically. And so then we thought, okay, well, can we load our matrix or our peptides at the molecular level with more chemical information that's specific to receptors? And so we took this RADA sequence, which is the self-assembling domain, and we added bioactive motifs. And again, we wanted to take a look at the effect on self-assembly itself. And so we can see from our AFM images that depending on the amount of molecule A that we put on, the size of these nanofibers start to increase, right? Even though they're individual still, they're just bigger. And that's primarily because we have more of a biomolecule attached to it. And then we wanted to see, well, can we actually use the chemical information that we programmed into the matrix as a means of inducing a cellular response? And so we were able to look at soluble fraction A versus tethered to the matrix fraction A versus just the bare matrix itself. And we saw that actually the tethering of molecule A to the RADA nanofiber induced a similar response as just the soluble molecule A. So that tells us right away, oh, okay, the morphology of the resulting structure is still good enough that the cell can read it. It can come in, the receptors can interact, the cell can get activated, and it can start doing its normal processes. And in fact, we were able to show a dose response effect as well. And it's really important to understand that this happened only locally. So only when the cell came in contact with the matrix does it actually respond. So we proved, I didn't show all the experiments, but we proved that there is no diffusion of something out, et cetera, et cetera. It was literally mast cell in solution. When it touches, boom, it starts its processes. So that's sort of a bioactive example of how we've been sort of thinking about how to mimic nature. Ultimately, what we'd like to see is some sort of a feedback mechanism, right? So For example, maybe we can activate the mast cell. Well, then in activation, the mast cell might release an enzyme. So can we use that feedback and information back to our matrix to release compound B that can maybe cause the mast cell to do a different function? Or maybe it can relate to a second cell type locally. And so to do that, this is sort of an overview of how we've started to build self-assembling matrices that are enzymatically responsive as a means not just of protein delivery or drug delivery, but rather as a means of building a communication network with the cells that reside within the tissue or within the matrix. In this case, we used a well-known protein, MMP2. It's really well known. The reason we really like it is because it's present in most cancer areas of active cancer. It's spatiotemporally regulated, meaning you can't really find it outside of the domain where it's being active. So it's regulated so it doesn't drift away from there and eat up other matrices and cause tissue reformation. And it turns out that this MMP2 protein can cleave these two different substrates with two different well-known rates. So it gave us an ability to see if we can form nanofibers with these two molecules, can we actually affect a different rate of release? And that's important because most things in the body are time sensitive. And then I have a whole bunch of pictures here. So this data was collected by one of the graduate students. He's just wrapping up now. He's done a really excellent job. But you can see the difference here compared to our other bioactive motif. You can see the appending of these constructs with time of sort of relaxing the system. You can see they have a radically different morphology internally relative to the control rata system itself. And in fact, you can start to see these nanofibers start to align. And so we started to analyze that. And it actually turns out to be a relatively good thing because the nanofibers themselves, when they bundle, allow cells to interact with them a little bit better. But how does this change in morphology affect enzyme access to these cleavable domains was the next question that's important. And through some relatively sophisticated mass spec work, we were able to detect and quantify the cleavage and product formation as a function of enzyme content. And we were able to show that indeed, despite these changes in internal morphology, the enzyme was still able to cleave these two substrates. And more importantly, the higher activity domains showed a greater amount of cleavage in the same amount of time. And so these were just in summary, these were just 3 or 4 different case studies to try to understand our approach when we start to think about how to mimic nature. We are interested in looking at peptide chemistry changes, how that influences nanostructure of the assemblies, and how that replicates, or I should say, and how that nanostructure further interacts with cell lines and how that affects their ability to do their job. And so none of this would be done without any of the students and postdocs. And I really thank them for their hard work, and of course all the funding agencies, which I forgot to put in, of course. Sorry. Anyway, okay, thank you.
Those are great presentations. I just wanted to ask Chantal a question, if I can remember it correctly now. So you were talking about the link to the land, and I was thinking about the diversity of First Nation, Métis, Inuit people, and even non-status First Nations, and how, I think those are really great ideas, especially around food sovereignty, but for some, for some communities, they don't have a link to the land. They won't have land-based, they won't be able to be land-based, even for Métis communities. So I was wondering your thoughts around that, because I was thinking in some of the work that I do, I think more around reclamation of identity, reclaiming traditional roles, as you were talking about, gender. I was thinking about that because I have Anishinaabe ancestry as well, and I was thinking, are there other ways to maybe connect those ideas? Just thinking, a little more broadly about that. And thank you so much for a great presentation.
Oh, thank you. Yeah, no, that's a great question. And I think that I have, as a geographer and someone who's really, you know, I'm from Northern Ontario, so my first and most formative experiences have been on Lake Superior. And my own experiences as a person who did not live on reserve, right, as a result of lots of complicated things, anyway, so I have this unique kind of history, and as I kind of had my experiences learning through university, have come to be an urban person. And suddenly I realized that the ways that I'm going to learn Indigenous knowledge and practice and teach to my kids is very different than, say, my sisters who do live in Northern Ontario and have access to elders and people and that land base. And so I guess this idea of repossession has some assumptions built into it that we may not necessarily have access to land in that same way. But it's okay, because the representations and places where we are going to be creating these new forms of indigeneity is not necessarily the same as in the past. And I think that's something that really needs to be talked about in Canada, because I think we have really old ideas about who Indigenous people are, not just in Canada, but, you know, globally. And so I think, obviously, I mean, I don't even know what the percentage is of you know, contested land and you know, the ways that treaties are you know, the contested land claim issue is hot. It's a hot topic, right? But for lots of different reasons, we are now a really urbanized population. And it doesn't mean that we don't have room and space to be exploring our Indigenous knowledge systems, but in new ways. And so I think that there's, you know, political, economic, cultural ways, and that's, those are promising places, but I think it's really it's unexplored. And I think to the detriment, because I think that we find a lot of kids, Indigenous kids, who are living in cities and don't know where their place is. And it's really sad. So that's why I'm hoping that we can explore that a bit more so that everybody knows where they belong.
Thanks. Yes, I've got a question for Patrick about photosynthesis. So I appreciate that you want to do it better than nature, but shouldn't the first step be to actually mimic nature and actually get some photosynthetic PV? Ever since I've read Ian McEwan's Solar, I guess, why aren't we doing this yet? What's actually stopping us from just grinding up blue currents, as you say, in a massively big scale and actually using it in a proper way?
Okay, I mean, that's a good question. Probably the thing that I didn't mention at all was, you'll see why, was that these molecules that I'm talking about will never be used in real solar cells, okay? Never. They are outstandingly difficult to make. Really, the kind of pinnacle of synthetic chemistry. You can make fractions of grams, milligrams, over month periods. Okay, so it really is a project of understanding at this stage, because we're never going to make these molecules. It's about what do we need to understand about this to make our own ones. So that I wouldn't say I glossed over it, but you know, the purpose isn't to make something in this project. What you're saying, of course, is, you know, really interesting, and actually the diversity. I mean, it was mentioned in a talk yesterday about the diversity of what's required in terms of generating energy from light, from people using pure just bacteria to create chemical energy, and people do this for even going to things like hydroponics. You need to define your problem and use the right solution to the right problem. So I think to get back to your kind of specific question, can we just take nature and use it to generate energy? It's not typically, at least in my awareness, it tends not to be so easy to directly extract it, and you do have to make changes. Also, a lot of biological proteins work very well in biological environments or physiological conditions, and that's not normally the same conditions you want to run a solar cell under. No, I hope that.
also have a question for Patrick. So when you show these rings and you say the words localization and delocalization, all quantum physicists will get really excited and think the thing you should be doing is putting a magnetic field through that ring somehow to turn localization and delocalization on and off. Do you look at this, or does anyone in your community?
So those exact molecules have been studied in this thing, and it's exceedingly difficult.
Okay.
Very fun. Yeah. I can talk to you after. It's one of these things that you would think you would see something really interesting. You actually have to basically put a charge on them permanently. So you have to dope them, and then they only last for a few hours. So you have a chemical problem. And then you have to run them at low temperature, and then you have to put them in a superconducting magnet, and it just becomes this kind of snowball effect of experimental horror.
So I thought I'd try to put a bridging question in here amongst this kind of very divergent, which I appreciate that there's a broad breadth to this panel. But I'm kind of curious about, one of the reasons we really worry about diversity in STEM is that we're worried about diversity of ideas. So ideas where things come from, different use cases that might come out of the technology. We see things like genetically modified foods in different cultures approaching that very differently as to the acceptability of that. And so I'm kind of interested in starting with Chantal, kind of trying to get a sense of how do you see some of these indigenous cultures in their, maybe not education, but their attempts to actually become parts of some of these scientific cultures to bring in those ideas and maybe bring them back or to tie them back in. And then kind of going maybe to the other panel to get a sense of how they feel that's actually happening or not.
I think that's a really great question, and I think it's something that's necessary and important for I think our economies, the global economy really, because I think that Indigenous peoples have been relatively excluded from the scientific community because our sciences are oral for the most part. And you know, the very nature of Indigenous knowledge systems means that we're not going to find a lot about this information in published scientific papers because it's knowledge that people are meant to know when they're meant to know it. So that's one part of the question. So we're talking about different forms of knowledge and the ways that you learn these knowledges, but also because Indigenous peoples have been relatively, I think, marginalized from traditional, and have not had equal access and opportunity to learn, you know, STEM or engineering. Even when our kids get into universities, they're traditionally kind of like pushed into First Nation studies, into social work, to go back to their communities and help in ways that are often crisis-driven. And I mean, there's like this really important revolution, I think, happening through the Canadian Institutes for Health Research, which has wonderfully funded, I think, myself, maybe Carrie, and developed this amazing group of young scholars who are leading the charge on Indigenous health and Indigenous health research. And it's starting to cross boundaries. And I think developing models of learning and success that are not just focused on Native studies, okay? And at Western, one of the very exciting developments has been the creation of this new interdisciplinary initiative in applied Indigenous scholarship. So there are a couple of really important, I think, political contexts for that. The first is that Western's in the process of creating our first ever Indigenous strategic plan, which is awesome. But also because we are trying to respond to the calls to action from the Truth and Reconciliation Commission, which was a multi-year commission into understanding the impacts of the residential school systems, which I talked about. And so there were a number of calls to action that were dedicated specifically for post-secondary educations and for the funding agencies. And part of that was to create more opportunities for Indigenous learners in our institutions and to make them more widely available and accessible and appealing options so that we don't just see, you know, Indigenous kids who get to university and choose these same options, but that they choose engineering and that they choose medical sciences and wide places. And I think there's a real opportunity for all of us to be a part of that, so that we can all see a role for ourselves in making our universities much more inclusive places for Indigenous learners and non-Indigenous learners. Because as we push forward in reconciliation in Canada, we're not just talking about making our country better for Indigenous people, it's for everybody, right? As soon as we start to, I think, reduce those inequalities that we see in Indigenous communities, we're all going to benefit. So I think that is a really great question, and I think that promoting interdisciplinarity for Indigenous ideas broadly, there's a real promise there.
Okay. Any other questions? Richard?
Did you want to respond?
Sorry, Patrick, did you want to come in on that? One sec.
Only because I feel I ought to. But actually, no, I mean, I find that this is coming from physics, you know. In many ways, physicists tend to think of themselves as having a monoculture. You know, there is one way of doing things and we do it this way, and it actually doesn't matter who you are or where you come from as long as you do it the way we do it. You know, I don't think that's true. That's what we'd like to think. But I was very interested with your answer, and actually to the extent at which we all need to try and understand how much we can gain by aligning the interests to try not to compartmentalize, because I think things tend to work better when we say, well, actually, you know, if we're, as a university, if we take the best people from anywhere, whoever they are, whatever language they speak, whatever culture they're from, we all gain. And whether you can do that without losing some of your culture is something that, I mean, it's outside physics, so I don't know. It's beyond my experience, but I thought it was a very interesting answer.
Okay, so I'm just going to put a view forward which isn't necessarily my own, but it relates to something that Patrick's been talking about. It's in photosynthesis. So nature isn't greedy in how it uses photons, right? So a tree will take up to 100 years to grow, and then we can take that tree, we can burn it and get the energy back in a very short amount of time. And what that shows is that it's actually not very efficient. So nature is sort of 3 to 6% efficient for photosynthesis. The best thing nature can do is kind of sugarcane, which is up to about 11% efficient. So if we're going to try and use nature, we're not going to do very well. I mean, we have silicon solar cells, which work at 20% and are much cheaper to produce than a field full of planting trees, for example, here, from an economic point of view. Of course, when a tree grows, it doesn't just create energy and store it for us, it supports the whole ecosystem. Right? And that's valuable in itself as a separate thing. So, two questions. One, is trying to learn from nature in terms of photosynthesis actually going to get us where we need to be for our energy demands as society? B, what about genetic modification? Should we be modifying nature to make it more efficient? Right? This is a controversial question I'm putting out here. And how do we also then deal with the fact that nature's also had millions of years to evolve, and it's kind of got to what you'd think would be an optimum point. So can it get any better, fundamentally? So just some thoughts there for people to try and think about and discuss.
Maybe I could dive in with the first part and then open the hard bit up to others. But you're saying about, what can we learn? I mean, is this the right thing to do, to be learning from nature? And I think I kind of made the point a little bit earlier that you're absolutely right. Nature is very, very good at doing what it wants to do. That doesn't necessarily, that's not what we want it to do. In a way, I do feel, although I have couched all my work in terms of photosynthesis, you can probably imagine it's to an extent, it is fundamental research about understanding. And I appreciate the point you make that actually we would have a long way to go if we tried to really adapt. We can do better using entirely synthetic silicon cells, or many of the other techniques that have been spoken about yesterday. In terms of, your second question was, should we modify nature?
Yeah, so nature is, we know it's quite inefficient. We have these great tools now, we have genetic modification, etc., as mentioned yesterday. So where are we as a society in terms of, if we know we can improve the efficiency of nature at growing, which might therefore make crop yields higher, you know, is that something, so that's just converting energy right into another source. Is that something we should be doing as a society? How do we address that issue? It's a more general discussion.
Which one of you want to take that one on?
Well, I think when it comes to energy production and light harvesting in particular, and we'll leave genetic modification for a second, but I think there's more to it than just raw internal quantum yields and stuff like this, right? And there's the ideas around product cycle, so cradle to grave analogies, recycling the materials, green chemistry. There's all these other things rather that play an important role, I think, on the justification between one type of device versus another. And of course, application plays into that as well. But so in some cases, if you can get something really cheap that only has 6% or 11% efficiency versus something that they put into outer space that reuses rare earth metals that cost $1 billion for that much, you know, I think there's some play between those two that can be found. And engineers should probably get into that, I guess. With respects to genetic modification, and I guess this sort of comes back to food as well. I was raised on a farm, and so my grandfather built his cattle herd literally from his dad, right? And his son took it over. That cattle herd was around for 100 years, roughly. And they selectively bred cows, right? They actually genetically modified their food and other things. So I'm not against genetic modification. The problem, I guess, with genetic modification that we should be worried about is the unintended consequences. So, and when it comes to genetically modifying bacteria or cyanobacteria to increase their light harvesting capabilities or to try to disrupt and maybe optimize light harvesting abilities, will we create a super cyanobacteria that takes over the world and kills us all and becomes the dominant species? I don't know, but that's my answer from a bioengineer's perspective. Thank you guys, by the way.
I mean, one of the things is I thought about during these talks is how we're mimicking nature ourselves, but how much does nature respond to the impact of human technological advances? So, you know, there's a two-way process here. So, as humans as we've gone on and pushed the technological boundary, I mean, how much is nature sort of starting to mimic us in certain ways? I know it's very difficult to measure many things over short length times, but do you know if there's any? Well, this is just me talking to myself. I'm not sure if anyone else will.
So I guess maybe what I'm getting from what your question is you know, these microbes do more than just harvest light, right? They store energy in chemical form. They obviously live, they replicate, et cetera, et cetera. And so I know like people are trying to use them as carbon capture mechanisms and things like that, if that's what you're driving at. So there is sort of a feedback between, you know, pollution and growth of molecule or of elements.
I think one of the things is, you know, we sort of put infrastructure in places that are now seeing colonization of species onto them, you know, that aren't naturally found in their environment. So how they're sort of, how they're reacting back to what we impact on them. That was the sort of point I was trying to get at. But again, it's just something that's going around in my head and everyone's looking at me blankly. So I will keep passing the box around. Do you want to just take it down there? Yep. Catch.
Okay, so this might be controversial, and also I'm not a scientist, I'm a humanist. But something that's striking me, I thought was great, the question about what the bridging question between the papers but something sort of fundamental is striking me, which is this discussion about us and nature, as though we weren't nature. And I think Chantal's presentation sort of underscores the fact that Indigenous knowledge understands us as nature, or as playing an integral role in nature, or being deeply connected to it. And so I guess I just wanted to hear people's thoughts about that sort of bifurcation between humans as sort of separate, the masters of all, the center of the universe, and consciousness and reason and all of these things, which I believe in, but also understand that old sort of Cartesian notion has been challenged, but also needs to be challenged, I think. That would be my claim. And I think, so when we talk about Indigenous knowledge, it's not just bringing Indigenous kids into the academy and teaching them how to be scientists. It's about scientists going to Indigenous communities and learning from Indigenous knowledge. So I just wondered if people had thoughts about that.
Yeah, who wants to? Patrick?
I completely agree with your question. And actually, I realized through my talk that the language I would use about nature, take, learn, or not even learn, take, mimic, steal, exploit. Right, that's the language that is used. And it's not, okay, I'm not saying, we're going to kill a load of animals and grind them up and stuff, but it is, there is a certain exploitative nature to looking at what nature does well and taking one aspect of that for something we want without a realization, and the point has been made that actually, trees grow slowly for a reason. If trees were 100% efficient, they'd grow in hardly any time, and they don't. And that isn't a question that I'm asking. I'm asking what's the best we can do. So, I mean, I'd certainly agree that it's not something that's rewarded necessarily in physics, my own field, to think about that. And it's certainly not something that if you said to, in a grant proposal, I want to do something that's more in tune with nature and less focused on absolute efficiency, it would be possibly a challenge to get that funded. Possibly, I couldn't say.
I think one of the obviously one of the big challenges is that we are academics, right? And our precisely what you're pointing out is that we are in this system where we've got to produce really quickly, and unfortunately, there's not often time to think about things that aren't directly related to your discipline. And I think what Carrie was trying to talk about yesterday, what you did talk about, is the nature and role of communities and Indigenous communities in guiding research. So this is one really new and exciting area, I think, of research happening in Canada that is gaining recognition and is going to gain, I think, a lot more recognition post TRC and so on by the federal granting agencies, is the role and responsibility for us to be thinking about the human dimensions of the work that we do, right? And that placing Indigenous communities, for example, in research is very important. So we held a panel last week and talked about our positionalities. So it was an interdisciplinary panel, and we're talking about the role for the disciplines in Indigenous research. And we had a biologist on board, and he's amazing because he's the sort of guy who's very interested in mercury in the environment, but is completely guided by his community partners. And unfortunately for him, he's working in NSERC and, the Natural Sciences and Engineering's research, right, which doesn't necessarily allow for the development of community relationships within a 5-year research plan, right? So we're often not afforded the ability to do those sorts of things so that we can accurately incorporate human dimensions into the applied nature of the work that we're doing. And I mean, the work that you all talked about yesterday, I was just sitting there and so confused, but also enlightened, because I'm thinking about, the pharmaceutical industry and thinking about mice and pushing buttons and thinking about how does this relate into everyday contexts. And it all has like important applications, but oftentimes we don't have the opportunities, I think, to bring this back to that human level. And I don't really know how we're going to get there, but I think it's one of the big challenges for us is to learn how to disseminate, and not just, it shouldn't happen just at the end of the research, but at the beginning, right? So how are we doing stuff that actually matters for overcoming the big challenges that we see?
I guess, if you don't mind, I'd like to
Yeah, no,
add.
please.
Yeah, I was challenged with this. I was a scientific advisor to curriculum development for K-12 in Alberta. And this idea of being able to communicate, like you were saying, Patrick, really came up. We were building basically mini labs, nanotech labs that kids could do in school to illustrate some different phenomena. And anyway, the point is one of the people who was hired to actually develop the lab and actually put all the pieces together mentioned trying to incorporate indigenous nomenclature into it. And it really challenged me individually because I thought it was totally a different realm of thought, right? Like, I could not really wrap my brain around what this person was trying to say to me about we were talking about microfluidic chambers and molecules and about the spirit of that. And this other person was trying to talk about the spirit of these devices and how to convey that in an indigenous context, which I think is what you're asking, right? And I found it, as a scientist, very confusing personally and challenging. And it took multiple conversations to sort of come at this discussion intelligently. Yeah.
Okay, just one question, just down there. Oh.
Thank you. I realize this is a science meeting, and some of us are social scientists, and we don't have linguists, for example, here, and I am not a linguist. But I'm struck by the languages we use. I recall a time, I've done some fieldwork in the Canadian Arctic, for example, and I realized after a full week of fieldwork that I asked 4 distinct questions in my mind, and the way they were translated in Inuktitut was always the same question, because there was only one word for all of that until my community partner pulled me aside and said, you know, we just use pollution for all of that. We actually use the word bleach because that's our closest analog. And then I hear Patrick talking about his reflection on his language of exploiting nature, and Chantel's challenges with a grandmother who learned to speak English. And then you talk about bringing indigeneity to urban areas. Does your language now have to evolve to work with the scientific community and with this evolution of indigenous knowledge and indigeneity itself? And if so, how? What guides that, or is it an organic process?
It's a really, it's a great question, and I don't really know is the answer. I think part of, so you're a geographer and I'm a geographer, so we understand each other, right? And geographers are pretty awesome because they seem to know a lot about a lot. I think we are like masters of all trades sometimes, or masters of, I don't even know how that works, so I'm going to stop now. But I think one of the greatest skills that I have is a social skill and ability to kind of talk to people regardless of where I am, which I can attribute to my mom, because she's my greatest teacher, but she is also very, she's like lights up the room. She's so bubbly and wonderful and knows how to talk to anybody and always makes friends. And this is, I think, one of the things that has guided me is that I know what I don't know, and am not afraid to ask questions, but know that I have to ask them in a responsible and respectful way. And I think that guiding, being led by that assumption means that I won't be offensive. Because I think that, well, hopefully not offensive, but I think that the ways that Indigenous peoples are changing and reflecting the new situations that we're in is very tricky, right? And I mean, when we talk about what what people are, who they are, how they identify, who belongs where, it's really contested issue. Because, I mean, Indigenous identity is a very complicated thing because it is a reflection of this silly political structure that has basically been imposed. So I think that people know who they are, and I think that you ask them to define what they think is important, and that's the best way to go about it. Does that answer your question?
Just, yes.
Mine is more a comment in listening to both the scientists and the social science aspects of it. One thing that has irritated me for 40 years as a biology professor is undergraduates that say things like plants, animals, and humans. And this is really one of the basic problems that we have, is in our educational system from a very early age, we don't teach our children that they are part of nature and they are an animal, whether they like it or not, in the true biological sense. And if we understood that a lot better, I think we would have a lot less problems and people would move forward on it in a much easier way if they accepted we are part of the system and what we do to the system will bounce back at us, and so we need to respect it. I mean, we have to move forward, but we move forward in a way that takes into account the ecosystem. But it means education from grade K, you mentioned it, from kindergarten up, that we are part of it, we are not separate from it.
So I'm not going to say anything as profound as him. The first point, I just would like to point out to Patrick that in the book Gulliver's Travels, that in the part that people don't usually read because it's the 5th book or something, that Gulliver visits a research institute where they are trying to distill the essence of cucumbers to bring the sunlight to winter. And this was a direct satire of the Royal Society. So you can see you're working in a good historical context. I feel that Chantal's point about language is something that is far more generic than the problems of Canada, because there are a huge number of minority languages, I think about half the languages in the world, that are in the process of dying out. And you can see already Chantal is not a native speaker of her own language. And the problem is that there may be things that in the small number of languages we have left, French, German, English, Chinese, we may not be able to think about certain thoughts. If you imagine the situation reversed, that Chantal's language was an ascendancy and French and English was dying out, the whole concept of gender would disappear, which would make whole areas of science much more difficult to do. And I suspect, but I don't know, that in these indigenous languages, they will have words for certain things, say in animal behavior and so on, which we don't have words to differentiate. As you know, that in some languages they have more words for colors than in others. So Russians can distinguish between light blue and dark blue better than the English, who it's all blue to. And so I just think that as well as looking at this problem from the point of view of the moral problem of the disadvantaged indigenous people, which clearly has to be addressed, we should also look at it at the huge opportunity for everybody to get some of the thoughts from these languages so that science can move forward. Because I think if you also put on a positive spin, everybody can see the advantage, rather than putting on the moral spin, you've been dreadful, now you've got to do something, which will tend, because of human nature, to produce a backlash and may not achieve what we really want, and make the indigenous people feel they have something to contribute towards the future, rather than just rectifying the injustices of the past.
To follow up on that, anyone?
So, Catch?
I just wanted to follow up on Jeremy's point about being part of nature and what Chantal was saying. Like when my cookum is giving teachings, my grandmother, she says there's two-leggeds, there's four-leggeds, right? There's the ones that swim, right? And there's ones with wings. So I think that's a really important point. And it goes to the idea that the two-leggeds were the last ones put on the earth, so we're the least important. And it's sort of a good teaching that when we think about, she always says, you know, if you want to think about it, Mother Earth doesn't really need the two-leggeds. And so if you want to think about, you know, preserving Mother Earth and being kind to Mother Earth, remember that Mother Earth can survive without us, right? And so just being aware and being cautious of walking softly on Mother Earth, being kind to Mother Earth. And you think about an ecosystem, you know, thinking about we are part of Mother Earth, we're part of that ecosystem, and you know, that's how I'm trying to raise my children as well, that we are two-leggeds, but also Mother Earth doesn't need us. So I just wanted to add that language.
Okay.